Thank you for having me. The details of my education probably aren't that important, but I see kids with disabilities. I see kids with autism. I see kids with brain injury, spinal cord injury, cerebral palsy, like bifida, all of those things. But I focus a lot on autism. I see a lot of children with autism because there's a lot of children with autism. So what I'd like to do today is give you an idea of the latest ways that we're thinking about autism from a scientific point of view. What's going on in the brain? What's different? What's different genetically? I'm not sure how much information I'm going to give you as far as treatment, but I think what I find is if I understand a little bit better how kids are thinking and why they might be doing some of the things that they're doing, it helps me understand and maybe make a plan. So I'm going to give you that background. I'm going to start though with, I think everybody has heard these numbers 1 and 88, the CDC data. And I wanted to let you know where those numbers come from because there's been, when I talk to people, there's been a lot of confusion about that. So our numbers right now are 1 and 88. The new data for the country for the next year, this data goes, we do it every 2 years. So the new data should come out next month. So be watching, we'll have new numbers next month. So it's a very costly diagnosis. In Arizona, we actually have data that are specific to our state. And only 14 states have that data. And I'll show you why. So we're lucky. Most states don't get to know what their numbers are. We actually know 1 in 64 and 1 in 40 boys. So where do these numbers come from? These are the 14 states that have been involved in the CDC project. Arizona has been in this project every year since the project started. The project started in 2000, and it looks at 8-year-old children. So when we looked in 2000, we were looking at children who were born in 1992. We were looking at 8-year-olds. The reason for looking at 8-year-olds is that if we look when kids are too young, we don't have enough data. They haven't had enough evaluations. They haven't had enough people observing them. So the way this happens is we can't ask for people's permission. Because think about it, if you've ever been asked to participate in a research study, you think about it, and it might be work, and I'm too busy. A lot of people don't sign up. So we can't ask people to participate. We have to go get the data. And the way that we do that, this is done actually in a big part of Phoenix. And the way that we do that is we send abstractors into doctors' offices, into schools, and they look at every child who's 8 years old that year, and they abstract every single evaluation that they've had, meaning that they retype every single evaluation, put it into a document. The reason they retype it is we can't copy school records or medical records. That would be a big privacy violation. So it's retyped with de-identified data. So nobody opts in and nobody opts out. And families actually, if you know somebody who's in this data set, we don't know who they are and you don't know that you're in. Okay? That's how we can be sure about the numbers, is that we're not asking people to sign up. So what happens is that the abstractors go in, they put together all of the data from the kids. It might be a medical information, it might be school information from wherever it is. And then they send it to somebody like me. There's 3 of us. And we code this paperwork line by line. And it'll say, during this evaluation, this child had poor eye contact. Okay? So that would be coded as a 1A, poor eye contact. Okay? And so we code all through this data. And at the end of the whole document, reviewing all the document, we add up all the codes. And if the kids meet criteria for autism, we say that they have autism. Nobody in those records, any of the evaluations, ever had to say that the child had autism. We just put them all together and looked at the data that way. So that's where these numbers come from. It's a paper-driven program. This is the data as it's happened since 2000. The 2010 data, the reason that you have delays is that it takes us a while to I mean, coding these things takes hours and hours, and then they have to be correlated, and then they have to be sent to the CDC from all of the different sites. So usually we're 3 to 4 years. It's every 3 to 4 years is when the last information comes out. So 2010 should come out, we think, next month. So you can see that the number of states reporting over the years has changed, but Arizona is in those every single year. And we're in it this next year as well. And you see that the prevalence, this looks a little bit different because our last one was 11.3 in 1,000. If you divide that out, that's where you get the 1 in 88. So we're looking at numbers per 1,000. So what's going on? We're going to talk about a couple things, identification, issues, changes in the brain, what's different, what do we know so far? We don't know everything. There's so much out there to be known. But what do we know so far? Some genetics and then a concept called epigenetics. Okay, who sees the arrow? Okay. If you're not looking for it, you don't see the arrow. Once you've seen the arrow, once you've been shown the arrow, you'll never forget. Okay? So I think that's part of it. Now, whenever you look at the FedEx truck, you'll see the arrow. And I think that as far as autism goes, that's part of the issue. We weren't really looking. And now we're very sensitive. Everybody else knows what autism is now. And we sometimes overcall it. But we're very sensitive because you can't miss it. It's right there. But you haven't seen it before. Actually on this, that the issue that I see a lot with that is that I do lots and lots of diagnosing. And I think at least half of the time, after we've diagnosed a child, one of the family members will call me the next week and say, do you see adults? Because I need you to diagnose my wife, my husband, my brother, or somebody. So there are many, many adults in our society who are undiagnosed. Now, if we had met them as children, we'd have easily diagnosed them. But we missed it. And we missed it, part of that FedEx thing. We missed it because we weren't looking for it. This is an interesting study done in South Korea. In Asia, there's a lot of shame to having this kind of diagnosis. So the numbers were different. And so people are saying, well, do they just not have autism there? What's going on? So they sent a whole team of autism experts and anthropologists to make sure that it was culturally sensitive and appropriate. We did this evaluation, went to Goyang, with a population of about 500,000 people, and they evaluated every single 7 to 12-year-old child. They screened them. If there were any concerns, they did a full evaluation. So with a really good evaluation in ADIR and ADOS, and the kind of assessment that we would do here. And the number that they got was 1 in 38. Not 1 in 88, 1 in 38. So I think that's what this really helps us. I don't know what the number actually is, but I think what it says is just autism is really common. Really, really common. Yeah, the interesting thing is that most of the kids had not been identified. They met all the criteria, but they had not been identified, and they were receiving no services. One of the things about that is that authors comment in this article that a lot of the kids were actually doing okay in school, and they think that they were because of how structured the school system is in Korea. It's very rigid, very structured, and a lot of memorization and rote learning. And so they weren't required to be creative, do a lot of creative writing, which is where they would have struggled. All right, brain. There's several different parts of the brain that we are very sure that are involved. And it's probably not, it's not one of these, it's the link between them. And some people might have a problem with one and not the other. Or they might have a problem in the whole system. So there's an area at the base of your temporal lobe called the right fusiform gyrus, it's also called the fusiform face area. I wouldn't expect you to let you know this. It's right in here. And what happens is that when you see a face, it lights up. Because people have a preference for faces over objects. We just like, even newborns. Newborns, if you put a picture of a face, put a picture of an object, they're going to look at the face. We have a preference for faces. And that's from the fusiform face area. People with autism, when you put them in a functional MRI and show them faces or show them objects, they light up about the same. Okay, so they're not really recognizing the face. Fusiform, or face movements. This is the right temporal parietal junction, which is right in here. What happens there is when you see facial movement, so you have a flat affect, and then I smile, or I frown. You go with my face, and you start recognizing different facial movements. Not so much, and the patterns that go with those movements. And then in your amygdala is a very primitive part of your brain. It's your gut reaction, it's your fight or flight. So if you're almost in a car accident, your heart rate doubles, and you automatically move. It's your amygdala that's making things happen in your body. So what happens is when you see somebody's face, your brain has an automatic emotional response. Good, bad, something. You respond to the emotion that's on a face. So you see that there's a loop there. We have to recognize a face as a face. We have to recognize the different facial expressions, and then we have to recognize the emotions that attach to them. This is the fusiform face area. And so this is, when they do these kinds of studies, this is what you see. They'll flash up, you're in an MRI scanner, and they'll flash up pictures of face, and then a table, and then a chair, and then a face. And they see when it lights up, when your fusiform face area lights up. So the control, that circle over here, it lights up when you see the face. The person with autism didn't light up. Then we get to, so that's just faces though, and social interaction is much, much more complicated than that. So there have been lots of studies just looking at how individuals with autism try to interact. What do they do? So this is a study, and there's several like this, I kind of like this one. They looked at Who's Afraid of Virginia Woolf. They had a movie screen. And they put headsets, visual trackers, on the people watching the movie. And all you do is watch the movie, but it's attached so that you can see where your eyes go. So they had people with autism and people who didn't have autism hang out watching movies. And this is what they found, is that the yellow is somebody who doesn't have autism. When you are sitting waiting for somebody to speak, we look right here. Because we're looking for facial emotions. We make eye contact. Because this is where you're going to see the emotion or of what's going on. Look for the person with autism. That's the red. Person with autism looks this and looks at the mouth. The problem with that is that you miss a lot of facial information, a lot of emotional information, facial expression, when you only look here. But you can kind of keep your mouth straight, but you're doing all kinds of things here. And then they looked at this part. This is a situation. The guy in the back is married to the woman on the left. The two in the front are flirting. Okay? So the person without autism looks, and there's this, it's called a triangle of interest. They keep looking at the guy in the back saying, wake up, your wife is flirting right in front of you. The person with autism completely misses it. Because they're listening, and they're going back and forth with the voice, but they're not getting the whole idea. So this person with autism, in this area, completely missed what was going on in this movie. So you can kind of think about how that might translate to social situations if you just don't get it like that. This triangle of interest is really important in human interaction. So we do this a lot. With little kids, for all of this, it's called joint attention. And in little kids, they do it, a 1-year-old starts pointing and grunting and moaning for the things that they want. Torture me. And then as you get older, it gets more complicated. And even you're trying to be polite, but you need to be someplace else. And you look at the person, and you look at your watch. You look at the person, and you're just trying to look, you gotta end, I gotta be someplace else. Okay? So that's called joint attention. So we do it in different ways as adults than little kids. Little kids, it's pointing, and then it gets a bit more complicated. So the next thing is on theory of mind. Theory of mind is being able to put yourself in somebody else's shoes. This is an evaluation that you can do by 5 or 6. Littler kids can't understand this, but by 5 or 6, most kids can do this. This is very hard for people with autism because it's a theoretical situation. So the top says, on the left is Sally, and this is Anne. Sally has a basket, Anne has a box. Sally has a marble, and she puts the marble into her basket. And then she leaves. Anne's just standing here, but Anne is very mischievous. And Anne takes the marble and puts it into her box. So, put yourself in Sally's place. When she comes back, where is she going to look for her marble? In her basket, right? Because she doesn't know that Anne moved it. But that's not the truth. That's not the right answer. The right answer is that it's actually in the box. So somebody with autism has trouble not knowing they're not supposed to know. They have trouble putting themselves in Sally's place and ignoring information that they're not supposed to know. This is a video clip on how we think some of this might play out.
Hello again. Gaze into a mirror and what do you see? Well, I see my face, of course, but in my face I see moods, I see shifts of feeling. We humans are really good at reading faces and bodies because if I can look at you and feel what you're feeling, I can learn from you, connect to you, I can love you. Empathy is one of our finer traits, and when it happens so easily, perhaps because, and this is brand new science, this is just out of the lab, we may have some special circuitry in our brains that helps us whenever we look at each other. Ask yourself, why do people get so involved, so deeply, deeply involved with such anguish, such pain, such nail-biting tension over football?
Cleveland Browns are gambling on defense.
Why are we such suckers for sports? And it's not just sports. We can lose it completely at the movies, at video games, watching a dance. Is there something about humans particularly, that allows us to connect so deeply when we watch other people? Watch them moving, watch them playing, watch their faces. Well, as it happens, scientists have an explanation for this strange ability to connect. It's new.
It had never been found on the cellular level before.
A set of brain cells found on either side of the head. Among all the billions of long branching cells in our brain, these so-called mirror neurons have surprising power.
What we found is the mechanism that underlies something which is absolutely fundamental to the way that we see other people in the world.
And it began entirely by accident at a laboratory in the lovely old city of Parma, Italy, where a group of brain researchers was working with monkeys. And they were testing a neuron, that's a brain cell, that always made this sound, whenever the monkey would grab for a peanut. So the lab had all these peanuts around, and whenever the monkey made its move, the neuron would fire. Scientists thought, now here's a neuron that is essential to motion. It's a motor neuron. Then one day the monkey was just sitting around, not moving at all, just sitting, when a human scientist came into the lab, and when that scientist grasped the peanut, yeah, the monkey's cell fired. Now, the monkey hadn't moved. It was the human that had moved, suggesting that this neuron up here couldn't tell the difference between seeing something and doing something. Seeing and doing were the same. Or more intriguingly, that for this neuron, watching somebody do something is just like doing it yourself. The head of the lab, Giacomo Rizzolatti, thought, wow.
The same neurons, one neuron, fire both when the monkey observes something and when monkey is doing something. It's almost unbelievable.
It was surprising because this cell which was involved with motor planning for the monkey turned out to be interested in the movements of other people as well.
Some people call them monkey see, monkey do neurons, but the name that stuck is mirror neurons, because with them, the brain seems to mirror the movements it sees. This accidental discovery got scientists thinking, doing more tests, and it soon came pretty clear that this is not just a monkey thing. It's a people thing, too. We all know that humans learn by looking and copying. That's what infants do. First you look, then you do.
Ready? Let's see your feet this way.
And once you've watched and copied and learned a set of moves, you not only have them in your head.
Look, you put your shoe on.
Yeah, she brushed it.
If you see somebody else doing it, you can share the experience.
And you want to do it with me?
They know the moves, you know the moves, so you can move with them.
Wow.
If you can use the years of training that you yourself have done, learning to crawl, then learning to walk, then learning to eat, this is an incredibly rich set of knowledge that you could apply to the problem of actually seeing what's going on.
So that's why when I head down this street carrying all these packages, not only do people watch, look how they're watching. They feel my predicament, because they know what it's like to carry heavy packages. They all know about carrying. So as they watch me moving, they can feel themselves moving. Their neurons are mirroring the action. These neurons may be the brain's way of translating what we see so we can relate to the world.
The mirror system is the way that you tap into, the way that you harness your own abilities and project them out into the world.
And people are really good at watching and translating what we see. Like with just 13 moving dots, that's all there are here, you'll have no trouble recognizing these very ordinary activities. What's more, tests have shown when a person sees a movie like this of his own movement, he'll recognize it immediately as his own. And that's why sports fans tense with the action and wince and leap, because if you know the game, then your neurons are firing as if it's you playing, giving a whole new meaning to the phrase armchair quarterback. That's why it's so easy to be a sports fan. But there is more, suggests UCLA professor Marco Iacoboni. He thinks mirror neurons tie us not just to other people's actions, but to other people's feelings.
So the idea was to try to figure out how the emotional system and this motor system are connected together. We're going to go in the scanner. What you're going to do is
to demonstrate, he put me into this very powerful fMRI brain scanner that can peer into the brain while it's working. And he gave me some goggles so he could show me pictures when I was in there.
So you can see here the eyeball of Robert.
And once he had a good view into my brain...
Nice looking brain.
Thank you.
Robert, you're not supposed to talk when we scan you, all right?
Sorry. Then he said, okay, I'm going to show you a bunch of faces. And for each face, I want you to imitate it. So I did that. Then he recorded my brain while I moved my facial muscles.
We're going to do right away another one.
Okay. Then he said, okay, same faces, but this time, don't move a muscle. Just look. So I looked. When we checked the results... Oh, there's my brain. I've never seen my brain before.
This is your mirror area.
Iacoboni says that the part of my brain that's working when I make a face, the same part gets busy when I see the face. Plus, when I was looking at these faces, I remember feeling extra uncomfortable, kind of bad. But when these faces came on, I felt better, almost happy. And in fact, at the moment I was looking at the happy face, my brain, and this is my brain, in that instant, see the red area here? It shows activity in the happy emotional part of my brain. And when I was imitating happy faces, look, I get even a bigger response. This, says Iacoboni, is a consistent result. Mirror neurons, he believes, can send messages to the limbic or emotional system in our brains. So it's possible these neurons help us tune in to each other's feelings. That's empathy.
We strongly believe that that's a unifying mechanism that allows people to actually connect at a very simple level.
You're saying that there's a place in my brain whose job it is to live in other people's minds, live in other people's bodies.
That's right.
Oh, darling, I'm going to die. Don't let me die.
Yeah.
And great actors instinctively know that if they put feeling and drama into their bodies,
Hold me tight. Don't let me go.
their faces,
It's dark out there alone.
we will respond.
Can't die. You're too brave to die.
What actors are expert in is using their movements to inspire feelings in the people watching. These are the experts in the mirror system.
We are intensely social creatures. We literally read other people's minds. I don't mean anything psychic like telepathy, but you can adopt another person's point of view.
When you put it together, what do you think it's going to be?
So if mirror neurons help us connect emotionally, what about people who have trouble with this? Kids like Christian, who has autism.
Why do you like Legos?
It's been known for some time that children with autism could be quite intelligent, but have a profound deficit in social interaction.
There he is.
Christian can speak and read and write, but like many kids with autism, he will avoid eye contact. He often misunderstands questions.
So, Christian, can you tell me what you did in school today?
Doing well.
You're doing well?
Everybody wants to know what exactly causes this. So Dr. Ramachandran and his graduate student, Lindsay Schenk, designed an experiment.
So we're going to be reading your brain waves with this cap.
They recorded brainwaves while the kids opened and closed their hands, and while they looked at a movie of somebody else's hands. For most people, the brainwaves look the same either way, whether they're doing or seeing. But for kids with autism, the wave changes, suggesting possibly that autism may have something to do with broken mirror neurons.
Their brains may indeed be different in that regard, and they may have deficits in the mirror neuron system. But we don't know this for sure yet, and additional work needs to be done using brain imaging.
But what we do know, says Ramachandran, is that healthy human beings are intensely social. More than our cousins the monkeys, we invent ways to connect. We invent dances and handshakes and games to play. We eat together, we meet, and we talk. We talk a lot.
Get away. Everybody's interested in this question. What makes humans unique? What makes us different from the great apes, for example? You can say humor, you're the laughing biped. Language, certainly. Okay? But another thing is culture. And a lot of culture comes from imitation, watching your teachers do something.
And here V.S. Ramachandran makes a big leap. He has proposed that at a key moment in our evolution, this is his guess, our mirror neurons got better. And that made all the difference, he says, because once we humans got better at learning from each other, looking, copying, teaching, we could do things the other creatures couldn't.
In other words, if you are a bear and suddenly the environment turns cold, you need a few million years to develop a polar bear type layers of fat and fur.
It would take many, many, many bear generations to select for furrier bears. But, says Ramachandran,
if you're a human, you watch your father slaying another bear and putting on a fur coat, skinning it, using that as a coat. You watch it, you learn it instantly. Your mirror neurons start firing away in your brain. And you perform the same sequence, complicated sequence. Instead of going through millions of years of evolution, you've done it in one generation.
And while no one's claiming that mirror neurons are the key ingredient that makes us different from other creatures, what these neurons do suggest about us seems almost self-evident. You can see it any Sunday at a sports bar, that deep in our architecture, down in our cells, we are built to be together.
There would be very little point in having a mirror system if you lived on your own. There'd be a lot of point in having a digestive system if you lived on your own. There'd be a good point in having a movement system if you lived on your own. There'd be a good point in having a visual system if you lived on your own. But there'd be no point in having a mirror system. The mirror system is the most basic social brain system. It's a brain system which there's no point in having if you don't want to interact or relate to other people.
But we do like to interact, and maybe now, as never before, we will understand why.
So, thinking about how we imitate. When we look at therapies for autism, what we're trying to do is teach kids empathy, teach them to use their mirror neurons, work on mirror neuron systems. And the therapies that we know that work actually do this. So I think of looking at therapies, the ones that focused on eye contact and listening and paying attention. It's usually ABA, applied behavioral analysis, very intense one-on-one therapy. And I think of that as getting ready to actually do something. The next step is teaching kids what kinds of boards to use, how to speak. Social thinking classes, practicing putting yourself in other people's positions. And then the last thing is, as kids get older, is what's the utility of this? Why do I care? Why do I want to be social? So there's layers of this, and a lot of therapies that we do really do tie back into the idea of a mirror neuron system. Genetics, autism clearly runs in families. I see a lot of families and 3 or 4 of their children, and they all have autism, and parents have autism. There is no single gene. We do have a lot of conditions that are associated with autism, but just because you have fragile legs, just because you have Down syndrome doesn't mean you're going to have autism, but you are at higher risk. This is a newer concept, and it's something called epigenetics. Maybe it's complicated. I find it very complicated to get your head around. And what it means, epi means on top of the gene. And one of the reasons that this came out, or became an area of study, was that in many conditions, you have identical twins, and one twin would have a condition like cancer or autism or something, and the other twin wouldn't. And these are conditions that we consider genetic. So how can you have completely identical genomes, and one has something and the other one doesn't? The way that we explain it is through this concept of epigenetics. And the methylation of histones I'm going to skip because I'm going to show you a quick video that really explains that. And the big question as you're watching this clip is to think about is, can you inherit the experiences of your grandparents and great-grandparents? One of the reasons that this came up was that the striking information that came out of this. This is the Hunger Games. It was during the German occupation in Netherlands during the winter and spring of 1944. And during this time, people who were trapped in Netherlands got no food. And they lived on 400 to 800 calories a day. So lots of studies have come out of the people who starved. And this is a nice picture. I have lots of pictures of really horrible looking people who were starving. This one study, there's others, but this study looked at women who were pregnant during the hunger winter. If you were pregnant early in gestation and then later got to eat, you had a baby with a normal birth weight, but that child probably became obese as an adult. And then the progeny further down had obesity, cardiovascular disease, and diabetes. And then the grandchildren and subsequent generations had trouble with dementia and other cognitive problems later on. Because your grandmother starved. And then mid to late gestation, these babies were small. And then the next generations, grandchildren, great-grandchildren, remained small. They didn't go back to their previous stature. How did that happen? So what this theory says is that what a woman does, the person, the first generation, she also does to her fetus when she's pregnant, and because the germ cells, the ovaries, the testicles, and everything are there in this, the fetus, the next generation. So everything you do, if you're going to have children, or when you did have children, you gave all of that to your children and grandchildren.
You ever notice that if you get to know two identical twins, they might look alike, but they're always subtly different? Yeah, whatever. As they get older, those differences can get more pronounced. Two people start out the same, but their appearance and their health can diverge. For instance, you have more gray hair.
No, I don't. Identical twins are the same DNA, the exact same genes.
Yeah.
And don't our genes make us who we are?
Well, they do, yes, but they're not the whole story. Some researchers have discovered a new bit of biology that can work with our genes or against them.
Yeah. You're heavier and I'm better looking.
Yeah, whatever. Imagine coming into the world with a person so like yourself that for a time you don't understand mirrors.
As a child, when I looked in the mirror, I'd say, "That's my sister." And my mother would say, "No, that's your reflection."
And even if you resist this cookie-cutter existence, cultivate individual styles and abilities like cutting your hair differently or running faster. Uncanny similarities bond you together. Facial expressions, body language, the way you laugh, or dress for an interview perhaps, when you hadn't a clue what your sister was going to wear.
Let's go.
The synchrony in your lives constantly confronts you.
When I see my sister, I see myself. If she looks good, I think, I look pretty today. But if she's not wearing makeup, I say, my God, I look horrible.
It's hardly surprising, because you both come from the same egg. You have precisely the same genes. And you're literally clones, better known as identical twins. But now, imagine this. One day, your twin, your clone, is diagnosed with cancer.
Por favor.
If you're the other twin, what can you do except wait for the symptoms?
I have been told that I am a high risk for cancer. Damocles' sword hangs over me.
And yet, it's not uncommon for a twin like Anna Marie to get a dread disease, while the other, like Clotilde, doesn't. But how can two people so alike be so unalike? Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Girdle. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones?
That's correct.
Because these are huge.
They're huge.
Can we weigh them to find out?
So if you take, this is...
Looks like they can barely walk.
They can't walk too much. They're not going to be running very far. So that's about 63 grams.
63 grams.
Let's look at the other one.
So it's half the weight.
Right.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time. causing obesity. Just look at this. So what accounts for the thin mouse? Exercise? Atkins? No. A tiny chemical tag of carbon and hydrogen, called a methyl group, has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.
In fact, it's the epigenome that tells our cells what sort of cells they should be. Skin, hair, heart. You see, all these cells have the same genes, but their epigenomes silence the unneeded ones to make cells different from one another. Epigenetic instructions pass on as cells divide, but they're not necessarily permanent. Researchers think they can change, especially during critical periods like puberty or pregnancy. Jurdel's mice reveal how the epigenome can be altered. To produce thin brown mice instead of fat yellow ones, he feeds pregnant mothers a diet rich in methyl groups to form the tags that can turn genes off.
And I think you can see that we dramatically shifted the coat color, and we get many, many more brown animals.
And that matters because your coat color is a tracer. It's an indicator.
That's correct.
Of the fact that you have turned off that gene.
That's right.
This epigenetic fix was also inherited by the next generation of mice, regardless of what their mothers ate. And when an environmental toxin was added to the diet instead of nutrients, more yellow babies were born, doomed to grow fat and sick like their mothers. It seems to me this has profound implications for our health.
It does for human health. If there are genes like this in humans, basically what you eat can affect your future generations. So you're not only what you eat, potentially what your mother ate, and possibly even what your grandparents ate.
So how do you go to humans to do this experiment when you have these mice and they're genetically identical on purpose?
That's right.
So who is your perfect lab human?
Well, then we look for identical humans, which are identical twins.
Twins. And that brings us to the reason why we're showing you Spanish twins. In 2005, they participated in a groundbreaking study in Madrid. Its aim? To show just how identical epigenetically they are or aren't.
One of the questions of twins is that if my twin has this disease, I will have the same disease. And genetics tell us that there is a high risk of developing the same disease, but it's not really sure they're going to have it, because our genes are just part of the story. Something has to regulate these genes, and part of the explanation is epigenetics.
Esteller wanted to see if the twins' epigenomes might account for their differences. To find out, he and his team collected cells from 40 pairs of identical twins, age 3 to 74. Then began a laborious process of dissolving the cells until all that was left were wispy strands of DNA, the master molecule that contains our genes. Next, researchers amplified fragments of the DNA until the genes themselves became detectable. Those that had been turned off epigenetically appear as dark pink bands on the gel. Now, notice what happens when the genes from a pair of twins are cut out and overlapped. The The results are far from subtle, especially when you compare the epigenomes of two sets of twins that differ in age. Here on the left is the overlapped DNA of 6-year-old Javier and Carlos. The yellow indicates where their gene expression is identical. On the right is the DNA of 66-year-old Anna Marie and Clotilde. In contrast to the younger twins, hardly any yellow shines through. Their epigenomes have changed dramatically. The study suggests that as twins age, epigenetic differences accumulate, especially when their lifestyles differ.
One of the main findings of our research is that epigenomes can change in function of what we eat, of what we smoke, of what we drink. And this is one of the key differences between epigenetics and genetics.
As the chemical tags that control our genes change, cells can become abnormal, triggering diseases like cancer. Take a disorder like MDS, cancer of the blood and bone marrow. It's not a diagnosis you'd ever want to hear.
When I went in, then he started patting my hand and he was going, your blood work does not look very good at all, and that I had MDS leukemia, and that there was not a cure for it, and basically I had 6 months to live.
Was epigenetics the reason? Could the silencing of critical genes turn normal cells into cancerous ones? It's scary to think that a few misplaced tags can kill you. But it's also good news, because we've traditionally viewed cancer as a disease stemming solely from broken genes. And it's a lot harder to fix damaged genes than to rearrange epigenetic tags. In fact, we already have a few drugs that will work. Recently, Sandra Shelby and Roy Cantwell participated in one of the first clinical trials using epigenetic therapy.
The idea of epigenetic therapy is to stay away from killing the cell. Rather, what we are trying to do is diplomacy, trying to change the instructions of the cancer cells, reminding the cell, hey, you're a human cell, you shouldn't be behaving this way. And we try to do that by reactivating genes.
The results have been incredible, and I didn't have really any horrible side effects.
I am in remission, and going in the plus direction is a whole lot better than the minus direction.
In fact, half the patients in the trial are now in remission. But while it may be easier to fix our epigenome than our genome, messing it up is easier too.
We've got to get people thinking more about what they do. They have a responsibility for their epigenome. Their genome they inherit, but their epigenome they potentially can alter, and particularly that of their children. And that brings in responsibility, but it also brings in hope. You're not necessarily stuck with this. You can alter this.
So thinking about that, we say, so clearly part of the increase in autism is better identifications, the FedEx phenomenon. But if there's also a true increase in autism, could it be a change in humans epigenetically? So people are looking into all kinds of different things. None of these have passed muster. We don't know anything yet. But these are the kind of things, how has our society changed in the last 50 years? These are some of the ways. So we don't know, but there's research going on all of these. Does anybody know what this is? So this Nancy's no. So this is Nancy's dad, and Woody Youngward flew this plane in 1949 for 47 days. And people all over Yuma, there were lots of people who helped them make this happen. Gave them food, gave them supplies, threw down things. So this is a part of Yuma history. And did that change the people who participated? Is Nancy different because of that, or her son? We don't know. Do the things that you choose to do. When you choose to do something one day and not do something the other day, does that change your whole genetic line? And that, I think it's a big question out there. So that's, when you're looking at things happening, that's one of the things to kind of watch over the couple of years. There's going to be a lot more information coming out. And there's a lot more to be done. Yeah.
In the Hungry Witcher, with the German occupation, didn't they also show that there was a doubling of schizophrenia?
Yes, mental illness in the progeny also increased. Mm-hmm. Yes. They also showed one good thing is that the people who were starved but weren't pregnant, actually, if they got pregnant after that, so they'd have this period of starvation, their children lived longer. So there's lots of things. So you could start maybe it's not good when you're pregnant, but it may not be so bad later.
Yeah, but doesn't make you stronger.
Whatever you
Exactly.
Right.
They're
stronger and they have stronger progeny.
So we have about 10-15 minutes for questions. We'll let you know when it's 10:02 because I know some of you are going to move on to a separate lecture. Dr. Rice will be staying in here. She's doing a lecture next on concussions. So go ahead and ask some questions. Raise your hand. If you could stand up so everybody can hear your question, that would be great.
She and I want to know, is there a way to reverse the epigenetics? Like, if in fact those external factors are affecting generations and future generations come, then is there that possibility that, like with the mice, that could be reversed?
Yes. So where that's so the question was, you ever hear that? So can you reverse this epigenetic problem? Yes, but you have to figure out what it is. And so there's a lot of research and actually treatments now coming out in cancer to reverse epigenetic changes in people who have cancer. And they are showing really good promise.
So when you have a child, like one with you guys, do you try to overcome that and say, look in my eyes, I'm talking to you, or do you say, okay, look in my mouth, I need you to go to your seat, I need you to get your backpack, I need you to do whatever it is. Do you try to overcome that and change them, or do you kind of speak to them and look with how they listen to you?
How hard should you push to make eye contact? So much of what people in the community, in the world, show on their face emotionally is up here, that if you do not look people in the eye, you're going to miss a lot. So if you don't force kids to make eye contact, your classroom situation might be easier because they're not so resistant, but you're really not helping them for the world because people in the grocery store are not going to be as nice as you are. So you have to be able to read faces in order to know how to respond to people. So it's something that kids really need to learn. They can absolutely be taught. Absolutely.
I work with ABA therapy with a child and I talked about it with mom, and recently she called me, she's in California, and she started chelation. Yes, and she's saying she's seeing a lot of improvement because she had a heavy system. So what do you think about that treatment?
So chelation Disparate is taking a chemical into your body to remove heavy metals. It's extremely dangerous. Our cells, our mitochondria, have heavy metals in them, and they're supposed to. And if you take them away, and if you take out particularly too much, you die. And there have been 5 children in the United States who've been killed due to chelation. So it's not a good idea. And the heavy metal testing, if you go to a real toxicologist who studies heavy metals, they will, I've had a couple of families who've done the same thing. And they come and I'm not sure exactly what lab they're going to, but they get these tests that say they have lots of heavy metals in their body. And then I'm scared that they're going to do this, and I send them to a real lab. And the real lab does not find any problems with their heavy metals. So there are some bad labs out there in this country. Hopefully they're using mild chelators, but the big chelators that are used kill people. What age are most kids diagnosed? Oh, so what age are most kids diagnosed? This is a huge problem. We should be identifying and diagnosing kids by about 2. The average age from that CDC data, we can figure out if kids had a diagnosis, when did they get it? In Arizona, it's 4.5. And so you may have an idea why it's 4.5. School. So I'm in the medical world, but the medical world has failed in autism. It's the schools who are finding and identifying kids. And so we could identify much younger. Most families, particularly if they're experienced families of other kids, they recognize it at birth. And so we could identify early if we were more sensitive to it. But at this point, most kids it's in schools that they're finally being identified.
I'm just wondering how late can autism onset? Like, how late can it begin? At what age?
We think it's from birth.
But then how old can you be until you're kind of safe?
When we're not going to...
Where you might need your chances of developing it are over. Like, I've heard you can get late onset autism. I don't know if that's true, but it was like age 8. So I'm curious, at what age does it, do you get, you're saying in vitro?
Birth.
Yeah,
birth. So
and it's only the diagnosis.
It's a recognition issue for the most part. There are a few conditions, particularly seizure disorder conditions, that come in about a year and a half or 2 that are associated with the developmental autism. But so if you have that medical problem that makes these symptoms, then possible. But no, but really, truly experienced parents, they tell me all the time, I knew that with this baby in the nursery, at the hospital, what they have.
I have a friend that has 2 autistic children, and it took like a year and a half for the diagnosis to finally come through so that she got services. Is there a better process?
I know the
when you're saying 4.5, she
tried, but she couldn't do it. So if you start earlier, the process does take a while, mostly because there aren't enough people on the medical end who can do it. There just aren't enough of us. And part of the reason, unfortunately, that there aren't enough of us is that, so I did a general pediatric residency. I could be a general pediatrician. I did 3 or more years working as, or training to be a developmental pediatrician. People like me in the community make half of what a general pediatrician does. So why are you going to do 3 extra years of school to make half of that? So people are not going into what I do. So they're going to get worse. Because child psychiatrists can do it, child neurologists can do a little bit of it, but the people who do the most of it are developmental pediatricians. And we're a dying breed.
I'm going to stop just for a minute, but then she we'll continue with questions. It's 10:50, so if anybody needs to leave to go to the next session, please go ahead and leave, but let's first thank Dr. Rice.